Hello, everyone. Welcome to BrainMap. This seminar series is co-sponsored by the P41 Funded Center for Mesoscale Mapping, housed in the Martinos Center. It is our great pleasure today to have Dr. Susan Whitfield-Gabrieli for giving us a BrainMap talk. Susan Whitfield-Gabrieli is the Tommy Fuss Endo Chair in Precision Psychiatry, the Associate Director of Center for Precision Psychiatry, the Director of Research for the Center for Comprehensive Healing, the Director of EPIC Lab at MGH, and a member of Harvard Medical School faculty. Dr. Whitfield-Gabrieli received her bachelor's degree in biophysics, and then pursued graduate work in mathematics and in physiology neuroscience at UC Berkeley. She had held research faculty position at Stanford University and MIT, and also served as professor of psychology at Northeastern University and founding director of Northeastern University Biomedical Imaging Center. Dr. Whitfield-Gabrieli's primary mission is to understand the brain basis of neurodevelopmental and psychiatric disorders and to promote translation of this knowledge into clinical practice. For this end, she employs multimodal neuroimaging techniques to investigate the neural underpinning of typical and atypical developments, as well as the pathophysiology of psychiatric disorders such as schizophrenia, depression, bipolar disorder, anxiety, and ADHD. Our ultimate mission is to discover biomarkers derived from functional and anatomical brain networks, which may be utilized for prediction of therapeutic response, early detection of mental illness, and precision network therapeutics, which with the hope of improving or augmenting currently available treatment. I would like to remind our audience to address their question at the end of the talk. Dr. Gabrieli, the stage is yours.
Thanks so much for that gracious and very thorough introduction. I think that's my whole talk, so thank you. No, that was fantastic. I wanted to say that first and foremost, I wanted to express my tremendous gratitude to all of you in the room here, many of whom are very dear friends, collaborators, and colleagues. Also to Susie and Bruce, who have directed this amazing center. I really feel that your continual, brilliant innovations and associated groundbreaking discoveries have been transformative in the field in general. That's really transformed my academic life, and I feel incredibly grateful to that. My lab and I are also very happy to move to MGH for the Center for Precision Psychiatry because it gives us the honor and privilege to collaborate more closely with all of you. So I'd love to talk and discuss potential ongoing, but also new collaborations. So a while back, there was a paper indicating that there's 100% increase in ER visits for children, very young children with suicidal attempts and ideation. And that was even before the pandemic. And of course, the pandemic spiked anxiety and depression in adults, but also children. And the New York Times a little bit ago just covered and flooded us with the information about that. What are we doing about this mental health crisis? In fact, the U.S. Surgeon General and leading pediatric organizations, both in the U.S. and internationally, have classified this as a national state of emergency in adolescent mental health. And so it's really upon us there's an urgent need to innovate personalized treatments that target really the core mechanisms underlying mental illness. So if you take a step back and think about, okay, well, where are we with neuroimaging and psychiatry now? Except for some rare exceptions, I would say that a lot of the papers that we collectively contributed to have been really interesting, and they oftentimes explore differences in psychiatric patients versus controls or predict outcome, but they haven't yet fundamentally altered diagnosis or treatment, except for, again, a few rare exceptions with maybe DBS and TMS. So if you think about it, well, okay, what are the critical barriers in preventing us from moving the mental health treatments forward? The four things came to my mind. One is that our mental health care system is still primarily based on remediation instead of prevention. Oftentimes we have experience-guided trial and error. Because we don't understand the pathophysiology of the disorder, we don't really know which SSRI or which drug treatment or behavioral treatment might work for which individual. And a lot of these treatments are ineffective in the sense that gold standard treatments may only be 30 to 50% effective. And again, the mechanisms are unknown, which is a critical issue. Of the 4 pillars of our EPIC lab, E stands for early detection. Can we use baseline imaging to identify brain-based biomarkers that might predict subsequent progression of symptoms? If we could do that, then we could intervene early and possibly mitigate symptom progression, possibly over illness. Can we use multimodal neuroimaging to predict treatment outcome and then tailor treatments in the name of precision psychiatry? 3, what I'll talk about mostly today is can we harness multimodal neuroimaging techniques that actually develop new forms of neuromodulation? And can we use these techniques like real-time fMRI neurofeedback to train individuals how to do other behavioral interventions like mindfulness meditation, cognitive behavioral therapy, or DBT when they're in the magnet, so they can actually watch their brain networks being modulated with behavioral interventions. And finally, can we use multimodal imaging to understand causal mechanisms? And so I'll just give you a few examples of what we've done in the past. So we've identified cortical targets through the use of intrinsic resting state connectivity to TMS, as many others have done recently with depression. And we also are looking a little bit more at GABA and glutamate. So if you combine MRS with real-time fMRI neurofeedback, you can identify how GABA and glutamate may be modulated with this intervention. With dosed Unger, we showed that the medial prefrontal cortex GABA concentration is systematically associated with an increase in default mode network anticorrelations. So that could be a useful tool to look at how it's modulated at baseline and with perturbation. And today I'll talk a little bit about how we can use real-time fMRI to trigger both experience sampling questions as well as trigger task stimuli to causally change human performance. And if you combine these together, understanding causal mechanisms may help us refine the treatments and innovate more tailored interventions. So thanks to you, Ken Kwong and Jack and Bruce and everybody, we have all of these neuroimaging like fMRI. And although we can look at brain structure and brain function, we can also look at the intrinsic structural or functional connectivity of the brain through diffusion, weighted imaging, and resting state. Today, I'll be talking primarily about resting state. So we know these are intrinsic, extremely low frequency fluctuations of the brain that cohere in the absence of any task, and individual variation, and importantly, intra-individual variation of these networks shed great light in brain health and disease. So I have an arrow here pointing out the default mode network, which can be elucidated both by looking at intrinsic low frequency fluctuations, but also in terms of task suppression. So if you think back when Mark Rakel made his seminal discovery of the default mode network, it's interesting to note that before we were using fMRI to identify regions of the brain that might be more active for a cognitive or emotional task. And so one might imagine that if you were to flip the contrast and flip the question, what regions of the brain are more active during rest when the subject's mental processes are completely unconstrained, then you might imagine that they're quite variable. But of course, Rakel and colleagues showed that the regions of the brain that are more active during rest relative to a wide variety of attention demanding tasks are remarkably similar, medial prefrontal cortex, posterior cingulate, lateral parietal, and hippocampus. And I say remarkably similar because in general, they're similar across individuals, but again, the heterogeneity or intersubject variance of that network, as well as the individual fluctuations, can be very informative in terms of how to intervene and when to intervene. So Buckner showed his beautiful paper and review paper indicating that the default mode network is relevant to disease. We have followed suit with Judy Ford, indicating that the hyperconnectivity and hyperactivation of this network is related to psychopathology transdiagnostically. And Mark Rakel himself had a review paper 3,000 papers after his initial paper and 14 years later, where he had a graph indicating all the papers that reference his first paper. And you can see what you highlighted here in blue is all the papers that actually referenced his default network in relation to disease. So some might even hypothesize that the contemporary mental health crisis is actually due to a disruption in the default mode network. Now, it sounds like it might be a bit of biological reductionism, but I actually believe there is a lot to that. The modulation of default mode network and its interactions with other networks may actually be very important in the mental health crisis that we're facing today. However, this network and all the resting state networks are plastic. So we can modulate this network not only by very invasive techniques like deep brain simulation, but even by lifestyle choices like exercise and mindfulness meditation. So if we take a closer look The default mode network here, we have a figure by Mike Box indicating this network being elucidated by just intrinsic resting correlations. And so here he has people lying in the scanner, places a seed in the posterior cingulate, looks at the functional correlations between every voxel in that region. And of course, you see MPFC and PCC highly temporally coherent. And those two core medial hubs are highly engaged when we're thinking about ourselves, as well as when we have no task whatsoever to do. So in that case, we're often engaged in some kind of mental time travel. Often we're mind wandering about the past or the future. And if we're in a healthy state of mind, we may be just reminiscing about the past or planning for the future. But if we're in a disturbed state of mind, like in depression, we may be obsessively ruminating about the past or obsessively worried about the future. If we have psychosis, we may be engaged in paranoid ideation or hallucinations, all of which are engaging these two core medial hubs as well as other regions of the brain. So we and many others have shown that the degree to which these core medial hubs are activated or correlated during rest is directly associated to more psychopathology in patients, in those at risk, and even in controls. So if you look at a closer look at Mike Box's figure here, you see that the regions in orange and yellow are negatively or anti-correlated to a task positive or to frontoparietal control network, which you'll see here in blue. And if you were to look at the time course of these two regions, you would, let's say the MPFC and PCC, of course, they are practically superimposed because the regions within the default network rise and fall in temporal synchrony. However, any region in blue, like the dorsal lateral prefrontal cortex, will rise and fall in a negative correlation. And that anti-correlation, we believe, is very meaningful in terms of cognition and mental health. So Michelle Hampson and we and others have shown that the magnitude of this anti-correlation is, in the very least, an indicator of cognitive performance. So we have a hypothesis that hyperconnectivity of the default mode network may be a targetable transdiagnostic mechanism underlying mood and psychotic disorders. Now, of course, even within a subject, it varies over time. And between subjects, there's a lot of heterogeneity. But if we can target this large-scale network, we may be able to mitigate the psychopathology that is associated with. Moreover, the two core medial hubs of the default network rope in, to put it colloquially, other regions of the brain that are specifically related to clinical symptoms of particular disorders. For instance, the MPFC is hypercorrelated to the superior temporal gyrus in psychosis, and that was first hypothesized by Georg Northoff. And also the MPFC is hypercorrelated to the subgenual anterior cingulate, which we know is a key hub in depression. So when you see these hypercorrelations, if you can get an intervention that targets the large scale network, not only can downregulate the hyperconnectivity and hyperactivation within the network, but you can also downregulate the hyperconnectivity between MPFC and SCG in the case of psychosis and MPFC and subgenual, and thereby downregulate symptoms like auditory hallucinations and internalization and anxiety and depression. We further hypothesize that another form of hyperconnectivity is just a reduction of the anti-correlations. So we think that reduced anti-correlations is also a transdiagnostic targetable biomarker of cognitive impairment. So specifically, if you look at the magnitude of the anti-correlation between MPFC and DLPFC, what we've seen is that not only is it related to cognitive performance in adults and children, but it grows or selectively grows during development. It peaks at young adulthood, and then it selectively, unfortunately for me, goes down in typical aging. So the magnitude of the anti-correlations are also decreased in many different populations that have cognitive impairment. We see this over and over. And the baseline anti-correlations predict the developmental trajectory of symptoms related to ADHD years later, even in an unselected pediatric population. Moreover, these anti-correlations can predict within fluctuations of mind wandering. So I'll show you an example with Aaron Kusi, where we did connect and predictive modeling within a subject to predict the fluctuations, a data-driven approach to predict fluctuations of these anti-correlations. And it was actually the MPFC, DLPFC that was the biggest feature there. We also know that this anti-correlation is related with GABA. So let's say this hypothesis is true. Let's say we have this hyperconnectivity both within and between networks. How can we modulate this hyperconnectivity? There's many, many different ways. You could go ranging from more invasive to less invasive. We could do deep brain simulation to the subgenual. We could do ECT, which actually then increases the DMN anti-correlations and psychosis and depression. We could do TMS to the region of the DLPFC that's maximally anti-correlated with the subgenual, which then decreases hyperconnectivity within the network. We could use ketamine to also decrease the hyperconnectivity or psychedelics. And we could use various antidepressants. Here's an example where an SSRI decreased the hyperconnectivity within the default mode network. So these are all ways in which we can decrease the hyperconnectivity of the network. But there's other ways, just simple lifestyle choices like running or exercise can also decrease this hyperconnectivity. And mindfulness meditation is excellent at downregulating this hyperactivation and hyperconnectivity. So I'll be focusing on that today. And with our colleagues here at MGH, I'll share a little bit of ways you can do it with ketamine, but also not only can you do this with mindfulness meditation, but you can also simply do it with deep breathing and other breathing techniques. And I'll tell you about the breathing technique that we're working with Ken Kwong, Phoebe, and Jude, who's here today. So there are many different ways to behaviorally and pharmacologically modulate this network. But in order to figure out how it's really modulating the network and how it's related to downregulation of symptoms, you need a lot of tools. And that's why I've been very excited to work with many of you to develop these tools. Our lab is on the intersection really of clinical translation, but we also do some development of tools, which we hope address the gaps and the barriers in the field and opens really new windows to understanding the brain network organization and function in both brain health and disease so that we can start to get biologically informed approaches to precision treatments. So here's an example of the ART toolbox, which we developed many years ago. And everybody knows this already, but that can be very helpful in identifying time points that exceed some specified limit or time points that have motion associated with it. And so in the field of resting state connectivity, this is critical because a little bit of motion downregulates the true correlation. So a lot of the papers that you read, especially with clinical populations and developmental populations, may be obscured by not effectively, which is now called scrubbing the data. So if you simply regress out, here's just an example of a 10-year-old child. If you look at the default network here, you can regress out motion parameters, but if you regress out motion parameters plus just a few time points that you've identified with this toolbox, you'll suddenly see the default mode network. So you're going to get different conclusions based on whether you regress out just motion versus the artifactual time points. And I think that's now very known in the field of resting state networks, but I think what's not known and what Randy Gallup and I are working on writing up is the importance of fMRI QA for task. And so that's something that we can talk about later, but I think that's the field needs to go there a little bit further. With Alfonso, we created a Con toolbox that addressed a lot of the issues in the field at the time. So we know that Byrne and Benatini in 2006 showed that a lot of these resting state networks are simply just due to respiration. So in order to get rid of that positive artifact, what he suggested was doing global signal regression, which of course was a fantastic way to decrease those false positives because the global signal went with the respiration artifacts and then you can get rid of these physiological aliasing artifacts. However, then Murphy and Benatini showed that, well, if you do this global signal regression, then you mathematically mandate these anti-correlations, rendering them uninterpretable. So when we started the Con toolbox, we wanted to come up with a different method of noise reduction, and we decided to do the CompCore method, where you take the brain, segment it into gray white CSF, and then regress out PCA components of this white matter and CSF. So the Con toolbox does both the motion correction and the CSF physiological aliasing. Over the years, we've developed a real-time neurofeedback system, and now with the help of Paul and Andre, we have continue to work on it. We have it now here at Martinos, which we're very excited about. Thank you, Paul. And that basically is a tool to do real-time fMRI neurofeedback. And finally, we're developing something called the Mind Balance Training Platform, which is a closed-loop platform whereby you identify biological antecedents to fluctuations and symptoms such as repetitive negative thinking, which would include things like rumination or worry. And once you identify those antecedents, you can trigger just-in-time adaptive interventions or safe flow states. But we need a lot of data. And so thanks again to my colleagues here. We developed longitudinal studies like the Boston Adolescent Neuroimaging studying anxiety and depression, which was really pushed forward with Anastasia and Bruce. And then we had, I was at MIT at the time, and then we have lots of colleagues at McLean, Diego Pizzigalli, Randy Auerbach, who's now gone to Columbia. And that's been a fantastic data set that many people have been using. It's open source. We also have a longitudinal data set with SHARP. This is a multi-site research effort to look at biomarkers for people at risk for psychosis. So we're looking at 300 clinical high-risk individuals, and this has been an ongoing long project with our colleagues in the Shanghai Mental Health Institute. So now I'll just give you a couple of examples of how we can use these brain imaging biomarkers for early detection. We know that major depression is a common debilitating phenotypically very heterogeneous disorder. And we know that there's hyperconnectivity in this network, especially in MPFC and PCC. And we've also shown in our bandit population that has teens that it's directly related to more psychopathology, that MPFC, PCC connectivity. And we know that the subgenual ACC is extremely important in depression. It's a hub, obviously, for TMS and DBS. Also has fewer glial cells, less metabolism. And recently there's a paper showing that it basically predicts suicidal ideation as well. So this is a very important region of the brain. And it turns out to be hyper-correlated to the default mode network. In other words, the DMN kind of ropes it in. So oftentimes you'll see this region of the brain as part of the DMN. That was first shown by Mike Grecius, but we've replicated that many times, and as well as in our patients who are teens.
So,
yeah.
You named that spot again.
Oh yeah, thank you. This is called the subgenual anterior cingulate. Thank you for asking about it, because I don't know any other disease that has one particular brain region where there's multimodal evidence that there's pathological indices that are important to look at, and it can also predict treatment efficacy. As Bruce Rosen has shown, if you look at DBS, you can look at there's four clinically relevant white matter tracks around this, where Helen Mayberg can stimulate and do her deep brain stimulation that improves treatment efficacy. It's a very, very important region. Thanks for noting that.
Yeah. Lisa, I ask a question. We see that all the time in breathing exercise. We always thought it's that hypothalamus, but not, but it's further out.
How interesting. Love that. So then you might think, is this a consequence of the disease, or is it an early predictor of the disease? So we looked at children who were at risk, familiar risk for developing depression, and we took the two core medial hubs, MPFC and PCC, and we looked at the resting state connectivity between kids at risk versus controls. And what we found was kids at risk already had hyperconnectivity within the default network and also to the subgenual ACC. This is not masked. This is the only region that came out, which is shocking to me. And moreover, not were they already hypercorrelated with no clinical diagnosis at this time, but the degree to which the MPFC and PCC were correlated to the subgenual directly correlated with CBCL internalization and anxiety and depression. Moreover, it predicted who was going to develop depression 3 years later, better than the clinical scales. So can we do one step further? That we're first, we're talking about patients, adults in patients, and we talk about kids in patients, and we're talking about kids at risk. What if we were to look at neuroimaging biomarkers in young children who had no clinical diagnosis and were not at familiar risk? They were not pre-selected. It's a normative pediatric sample. So we tried to look at that. It's a longitudinal study, 94 typical children. We looked at the CBCL, the child behavioral checklist, and then we scanned them every year for 4 years and took behavioral. Well, we did pre-register this, and we pre-registered that weaker functional connectivity between the subgenual and DLPFC might predict worsening of depression. And so that is indeed what we found. So what you'll see is that, but we wanted to basically replicate that. So if you took that same model, the subgenual ACC and DLPFC connectivity, in a different population, now we're looking at what goes on there in children at risk for depression. It also predicts worsening of anxiety and depression 3 years later. We wanted to replicate it one more time. So then we took it to our teens who are already identified as having anxiety and depression. And again, the subgenual ACC and DLPFC predicted worsening of anxiety and depression and internalization there as well. So it's possible to wait till you're an adult to get TMS and get targeted and target that area in the DLPFC that's most anti-correlated with subgenual. Or I've shown you 3 different populations where the same connectivity is predicting worsening of anxiety and depression. Here's 1 year later, here's 3 years later in children at risk, and even in a normative population. So is there a way to identify, and you don't even have to identify children if you wanted to just, put the fluoride in the water, to basically give people something like mindfulness meditation in the schools that we know will downregulate hyperconnectivity, the default network, and subgenual, and increase this anti-correlation. So in schizophrenia, we looked a lot of people who were first episode or familiar risk, and we found hyperconnectivity of the self-reference nodes again, and this related to positive symptoms. We've replicated that in much larger databases now. And if we have looked at many different samples across the age span for children who have familial risk, children, we have a few hundred people who are clinically at risk. We look at chronic schizophrenia as well as people who are schizophrenia, but also have co-occurring cannabis use disorder. And so across the lifespan, what we see is this hyperconnectivity, and the biggest really group difference is that there's a lack of anti-correlations, which we think there's a lack of cognition. If you look at people who are having cannabis, then that increases. If we had a bong in a scanner with dark meth, and we actually gave these people cannabis, and we saw the increase in the anti-correlations that was going with an increase in working memory performance. So you can see it's directly related. So let's just turn to precision psychiatry for a moment. Precision medicine is defined to be an emerging approach for treatment and prevention that takes into account each person's variability in genes, environment, and lifestyle. And of course, there's many things that we can measure here. And then we can use systems biology and computational psychiatry to really get this biosignature, which then can be useful for treatment in psychiatry. Randy Gullip had a beautiful symposium, and she wrote a great paper of how machine learning can empower neuroimaging to improve mental health. And Leanne Williams has done a lot of this work on precision psychiatry, looking at imaging-derived phenotypes to predict outcome. So I'll just give you an example here where we can look at how we can use these multimodal imaging technologies that you all have developed and inform clinical outcomes. So here's an example of where we knew that the subgenual ACC DLPFC connectivity would predict worsening of internalization, anxiety, and depression. But as Bruce has often shown, and Helen Mayberg has shown, that we also know from diffusion that if you target subgenual ACC on the intersection of those four clinically relevant white matter tracks, then you can get better treatment efficacy. So why don't we harness that information and use that to get better predictive power? So that's what Jiahe Zhang is doing in our lab. She's looking at the diffusion and then combining the diffusion with the resting state intrinsic connectivity to get a better predictive model. You can also use this baseline imaging to predict treatment outcome. We've done this with people with social anxiety disorder, where we've given them a task and they can look at fearful greater than neutral faces. And what we've seen is that combining the brain measures of that activation to the initial LSAS, then we can get better predictive power, and we can look at, we basically accounted for 40% of the variance in treatment response, which substantially exceeded predictions based on the clinical measures. We can also use intrinsic connectivity here. We can use a data-driven and a hypothesis-driven, hypothesis being amygdala connectivity, data-driven meaning MBPA, and we can combine that with intrinsic connectivity of diffusion, and that gave an even better response. And that was a 5-fold improvement when you combine the intrinsic connectivity with the initial LSAS. So there's a number of ways we can use neuroimaging to predict treatment response. So now the neuromodulation. So we know that there's many different ways to modulate the networks. One is simply mindfulness. We know that mindfulness downregulates the default network, increases cortical thickness, increases GABA, and improves many clinical symptoms transdiagnostically. It also, we've shown that it increases educational outcomes in children, and also importantly, decreases self-perceived stress. We did an RCT right here in Boston where we gave people mindfulness, 8 weeks of mindfulness, and we brought them into MIT to look at the brain changes before and after. And the brain activation to amygdala significantly decreased after this mindfulness, 8-week mindfulness session relative to a scratch programming active control. And the degree to which the amygdala downregulated corresponded to a significant downregulation of self-perceived stress. And when we looked at the resting state connectivity, we saw a significant increase in the anti-correlations, which was associated with an increase in sustained attention. So we can see in kids and 6th graders that mindfulness actually modulates these intrinsic networks as well as anxiety and sustained attention. And now more and more people, this is not my work, but some people who have come from MGH have shown that mindfulness-based stress reduction can be just as good or not inferior to SSRIs to treat people with anxiety, and it can be even better to treat people with depression in certain cases. So it's becoming more and more known to be a fantastic treatment. But there's no feedback. So this is where we get to the real-time neurofeedback. We started doing this way back when in Stanford in the early 2000s when Christopher DeCharms came to us, and we wanted to know, okay, typically we're usually manipulating the mind with some kind of fMRI task. We look at the brain activation, and publish it in Science. But now can we reverse engineer that whole thing and look at the brain activation and ask, can people learn how to modulate those regions? I think many people now believe that's true. But the more important question is learned regulation of the activity or connectivity, does that learned regulation actually influence the mental processes that's related to? And I think many people would say yes. Randy and Eden Evans and Luke have created a really nice symposium at one point, and this is a nice paper that came out of that, illustrating the clinical utility of real-time fMRI neurofeedback. And one of the papers that I think would be most impressive is that Alison Alcock and her colleagues showed that one single session of real-time neurofeedback where people can actually look at how the brain is modulated with things like emotion regulation or reappraisal improves CBT strategies subsequently. So if you look at over the course of time, there's been kind of an exponential increase since the first paper that Chris DeCharms showed, and it's being taken more and more seriously by NIH. So right now, what we're doing is not modulating a region of the brain. We're trying to modulate a whole network, specifically the default network and its interactions. And so the way this works is we have people lying, we first train them to do mindfulness meditation. We have them lying in the scanner. We quickly analyze the data, and then we feed back the signal in the form of a ball test, which I'll explain a little bit more. But the idea is that we think that the ability, training people to modulate this large scale network as interactions might be a better way for neuromodulation and more effective than just looking at a single region. And it may be better than involving just an unspecified pharmacological intervention. So far we've had a lot of success with this. And this is the way, it's also very personalized. We get the individualized networks. We meet them where it's at. So if they can't do it, we'll change the hop size of the ball so we can actually get them to move it around. And most importantly, this intervention empowers these patients with agency, and that's definitely what they like the best. So rather than being a recipient of DBS, TMS, CBT, or DBT, you're actually you're learning how to modulate your brain and subsequent behavior in real time. So here's what it looks like. We have people lying in the scanner. We immediately get their personalized intrinsic networks, and then we track the default network and we track the frontal control network. We convert that signal into a ball game. This is what the patients are seeing here. And the idea is that they're going to be passively doing mindfulness meditation that we've taught them, and occasionally looking at where the ball is going. The feedback is such that if the ball is moving up, you're successfully doing mindfulness meditation, and the default mode network goes down relative to the frontal parietal. But eventually people tend to mind wander, and then the default network starts to rise relative to the frontal parietal, and then the ball moves down. So basically they're just wiggling this ball by doing mindfulness meditation, and they're learning how to train the brain to tune their mind with mindfulness, because they're learning when they're pushing down, doing mindfulness is helping you focus on the present moment rather than rehashing the past or focusing on the future. And that present moment orientation allows the downregulation of the default mode network relative to the frontal parietal. That's how they can move the ball. Yeah.
So basically, once they get that feedback, what are they supposed to do? It's like focus more on your meditation, or what do they actually do?
Well, that's a great question. What do they actually do? So I'm just let me go back and show you exactly what they're doing here. So they're just trying to get into the circle on the top. And when they do, the circle shrinks to make the task a little bit dynamically more difficult, but they're just playing a game, and they're just playing this ball game. They're just trying to get it up as much as they can to get it up into the circle. So right now you can see that they're moving the ball. They start meditation here. You can see they're down regulating the default network. So the ball is moving up. So this is an example of somebody who did it well.
Well, I asked because like in motor imagination tasks, it's very clear. You try to focus on your right or left hand.
Yeah.
But in this, what are they supposed to like meditate better, or what are they actually doing to make that brain activity change and then see it?
Right. So what we teach them outside is a type of mindfulness meditation called mental noting. And so they can try various forms of that, but basically they're doing the mindfulness meditation and with a mental noting, and they're trying it. And if it seems like the ball's not going in the right direction, they may do it with a slightly different strategy, but they're basically trying to do that overarching meditation until the ball goes up.
So they try different strategies to see which one's bringing the ball on.
Yeah.
So the mindfulness, is it like mindfulness to breathing or heart rate or anything?
Yeah, so that's a great question. So there's so many different ways to do mindfulness, and one way to do it. It's just simply focus on the breath. And so that's another way to do it. We do, that may be, the more you focus on your body, sometimes that can be triggering for people with trauma. There's a number of different reasons why we actually chose a slightly different mindfulness meditation, but the breath work is an excellent way to go as well. I think I just, oh, no, okay. So yes, breathing is a fantastic way to modulate. And as you know, we can actually modulate the networks quite well using various breathing techniques. But then we want to also know how the intrinsic networks are modulated. So we do resting state before and after. And what we've shown is that we significantly decrease the hyperconnectivity of MPFC and PCC in individuals with psychosis. So they have hyperconnectivity at the beginning, but that is downregulated after this intervention. They don't have any anti-correlations between the MPFC and DLPF at first. That's a 0 bar. But then after this intervention, it pushes down. In other words, it increases the anti-correlation between MPFC and DLPFC and MPFC and dorsal ACC. But with motor movement, that's our sham. You don't get that modulation of resting state networks. Now, again, this is just one session. If we did this over and over and over, you probably would, just because you're still doing mindfulness meditation. So I'll just show you a couple of examples here. We're targeting STG. We're looking at STG and having individuals with psychosis focus on their own voice and ignore other people's voice through mindfulness meditation. When you do that, you can see that they're downregulating the hyperconnectivity. This is each person's effect size. And that happens when you're feeding back the real activity in the STG versus the sham. And the same thing is that you get reduced DMN STG connectivity when you do this intervention. Remember, the DMN is hyper correlated to the STG. And that reduction is what goes significantly down with the hallucinations. So we're very excited about using this for individuals, especially teens with depression, to treat RNT, or repetitive thinking like rumination. And so we brought back our BANDA subjects many years later. This is a pilot study, but basically we looked at 9 subjects and we showed them the mindfulness neurofeedback, and they were all able to downregulate the MPFC and PCC, and they were able to downregulate the MPFC subgenual anterior cingulate, which is really the key thing that's related to internalization and anxiety and depression. And we found that increases in state mindfulness, which they had, was associated with a reduction of the subgenual ACC MPFC connectivity, and that the reduction mediated the association between where they have better performance and increased state mindfulness. So basically, we think this is a transdiagnostic intervention that downregulates this hyperconnectivity within MPFC and PCC. It downregulates hyperconnectivity between MPFC SCG, and it downregulates hyperconnectivity between MPFC and subgenual, and it increases the anti-correlation. So we're really excited about this large-scale network modulation.
How long is the effect?
That's a great question. So now we have about 6 clinical trials, and we're actually asking how long does this effect. Michelle Hampson has shown at Yale that the one session of real-time neurofeedback actually has long-lasting clinical effects, well over a month. We have not shown this yet because we haven't had the longitudinal data, but we're collecting it now. But the good thing is that we think that the real-time neurofeedback might not actually in itself be the trick to the intervention, but it's actually just training wheels to train people how to do the mindfulness meditation. So even if that particular session didn't result in long-lasting clinical benefits, if they were able to learn and it augmented the skill acquisition and utilization, then the actual mindfulness meditation that you do afterwards would probably have a long-lasting effect. Yeah.
So how easy can you identify inside the scanner? Because STG is like involving the audio.
Yeah, so SCG is very auditory. It's exactly right. So yes, so we worked really hard on that. So we had to get headphones so we record their voice and we record somebody else's voice saying sentences. And so we have to work very carefully so that they can hear their voice versus somebody else's voice in that loud environment. But we're able to do that. And so it's a differential between hearing themselves and focusing on that versus hearing somebody else's with mindfulness. So that differential is what causes the decrease in connectivity. Yeah. No, but that's another way. Some people also do that where you can actually break and do it, but this is actually during the scanner. Okay, so I'm just going to skip forward. What time is it now?
42.
Okay, so I'm just going to be quick here. So now we're trying to use this mindfulness neurofeedback on patients with schizophrenia longitudinally, but we're also trying to augment DBT and borderline personality. So, dialectical behavioral training has many modules, but the first of which is often mindfulness meditation, which has been shown to be better than interpersonal effectiveness in terms of downregulating symptoms. So what we're doing with Yale University is trying to use real-time neurofeedback to increase the skill acquisition and utilization of that particular module versus not having it. So that was with individuals with borderline personality. We also have a number of projects where we're trying to capture the electrophysiological correlate of the default mode network and its anti-correlation so that we can scale the intervention. And we're now here, we're just setting it up at Martinos, MRS, where the boxes are functionally localized with intrinsic resting state networks, so that we can look at that GABA and glutamate and MPFC and DLPFC. That was motivated based on our finding with dose longer that MPFC GABA is increasing with MPFC DLPFC anticorrelations. So if it's true that these interventions are increasing the anticorrelations, then we should see increase in GABA before and after the interventions. With Columbia, Randy Auerbach, we're looking at a large scale RCT to see if we can mitigate rumination with this technique. And hopefully we'll still have this grant in China, but with our colleagues in Shanghai, we're looking at TMS because of Roscoe Brady's finding where 2 times a daily intermittent theta burst of TMS modulates a cerebellum midbrain VLC connectivity and reduces negative symptoms. And so we're going to see how that works in comparison to the mindfulness-based neurofeedback. So I think what would be really fun to strategize ways in collaborating here with people who are doing interventions might be to combine various interventions to get synergistic effects. I've told you that you can do mindfulness inside the scanner, but you can do many other behavioral interventions, and this will augment your agency to learn how those strategies are working. And then you can also prime the neural circuitry that you want to subsequently modulate. So you can have people, we've had kids come to Northeastern and exercise first to increase their anticorrelations, and then we modulate them. But you can also imagine doing something like mindfulness meditation right after ketamine. We know that the ketamine induces this neuroplasticity window. And if we can have an opportunity to inject a behavioral intervention during that window of plasticity, we may be able to have longer clinical benefits. I'm going to skip that. So oftentimes baseline connectivity predicts treatment effects, but what we're really excited about is looking at dynamic biomarkers or perturbation biomarkers of brain elasticity. So there's one paper showing that if you look at change of resting state networks before and after, 5 hours before and after an SSRI, that predicts long-term treatment efficacy of depression, which was to me shocking, but a really interesting methodological point. Can you use a change in biology, like the change in resting state networks, to predict long-term treatment efficacy? It can be anything. It can be any kind of brain perturbation, TMS, DB, whatever perturbation you want. Resting state networks that you guys came up with fMRI can actually be used to just measure the perturbability, malleability, plasticity, or adaptability of the brain, and so that you may be able to get predictive biomarkers, prescriptive predictions that may be able to say, okay, this person is better for a top-down intervention. This person is better for a bottom-up intervention. So for instance, if we're doing a bunch of ketamine, real-time neurofeedback, TMS, exercise, can we create a structure such that we do resting state before the intervention? Of course, with real-time, we can do it during too, and then resting state right after. So we get a perturbation index that's reflective of the malleability of the brain to that particular intervention. Then we let the intervention go on and we get another resting state network. This can be combined with EEG, all the heart rate variability, everything like that. So you get a suite of biological measures at the baseline, right after the intervention, and then at the end of the intervention. The baseline and the perturbation index, the degree to which the body and the mind change, will then be, I think, a prescriptive predictor that could be perform the baseline predictors. So we know that the resting state during rest, it's not like we're task free. We know the sensory information from the body and the world is perturbing our brain dynamics all the time. So one hypothesis is that some people can get locked in, for instance, locked into the default mode network. And this may relate to depressive disorders, which we know are also combined with metabolic illness and may be caused by dysregulated interoception and neural inflexibility. So we're trying to look at that with our colleagues here. And Susie first suggested it, where we're going to look at multiple imaging sessions, 3 time points, where individuals with depression come in to the MGH clinic and get ketamine. And then we're looking at the change of resting state networks at 3 time points. And what we're seeing here is that if you do a data-driven approach, an MBPI approach, to look at, okay, how has the brain connectivity changed over 3 time points? What was amazing to me is that it was precuneous, which we know is kind of the hub of the consciousness, right? It's a posterior region of the default mode network. And independently, she did an MBPA on what's changing with brain in relation to the change of symptoms. And she got pretty much the similar case. And so if you look at the network connectivity changes over time, initially what happens along with many psychedelics is that you increase, even though ketamine is not a psychedelic, but basically you're increasing the connectivity amongst many large scale interactions. But then if you look at the next, you can see a decrease in MPFC and PCC. And now we're doing a really exciting study with Jude, who is working really nicely with Ken and Phoebe. We're very grateful for all of their expertise. And now we're looking at dynamic volitional respiratory modulation. There's been a number of papers coming out showing that different types of breath work can actually induce psychedelic experiences, which can be actually very helpful for people who are in trauma and many other psychiatric disorders. So there should be some pilot data illustrating the increase in the large scale networks initially, and then the modulation of the default network thereafter. It's still in process. So I would like to suggest that measuring this neuroplasticity or malleability may be, in the long run, a really good way to go clinically. So we know that in cardiology, we know that we want to image the heart as an organ of interest. We look at the blood flow during rest and during stress. Why can't we look at the brain? Just bring everybody into the fMRI and image the brain and the organ of interest, and look at the blood flow during rest and with various cognitive and emotional tasks. So even with that ball task I showed you, if you push the brain up and down, you can get kind of like a VO2 max indicator of the malleability of the brain, which might be really helpful to then figure out which treatment that person should be getting. Okay, so I have a whole nother part that I'm not going to have time to get to, but I will just skip to one particular study that I think is important. And that is here. So we talked a lot about looking at individual differences and predicting outcome, but I think really where the field should and could go is to expand the lens and look at deep phenotyping individual variations, because the variance in the individual fluctuations can actually be greater than between subject variation, and we ultimately want to treat the patient of interest. So here we did predictive modeling of mind wandering from functional connectivity. This was with Aaron Kusi. We had a number of data sets where we created the model. We had experience sampling. And what we did was we had training data, and then we had mind wandering ratings, and then we looked at the Schaefer Atlas, and we looked at the 30 seconds prior to that rating. And we basically modified the code, the connect and predictive modeling, which you've heard about in BrainMap from Todd Constable, that can be used to look at individual between subject variations and behavior and symptoms. Here we modified that code to look at the changes within a subject. So we're doing a data-driven and CPM approach to look at the fluctuations of mind wandering within a subject and see if we can capture the data right before the experience sampling to see if we can make a predictive model to predict when somebody's going to be mind wandering. And so we were successful in that. We were able to predict with a data-driven approach. And the two features, the top features, were the MPFC DLPFC anticorrelations that predicted. We also saw decreased coupling between the default network and sensory motor regions, which may be a process that underlie perceptual decoupling. So we replicated this in many different data sets, large-scale data sets. But the idea here is that this is a proof of principle approach that can be applied broadly to develop many different predictive models within a subject that could predict future fluctuations, suicidal ideation, rumination, or what have you. The key thing, though, that I want to point out is that even if we assume, let's say, the default mode network is, if we can track it when it's high, we don't know specifically where they're going when they're mind wandering. Detecting mind wandering is great, but what are they doing when they're mind wandering? So we developed a real-time triggered experience sample protocol where we track the fingerprint of interest, and then we trigger questions. So what you see here is the difference between the default network and the frontal parietal. So we can tell, or we think we can tell, when the subject's mind wandering, and then we'll trigger it with this blue arrow here, and the subject responds, yes, I'm mind wandering. And then we'll track, again, the difference between those networks. And then we'll say, you're on task or off task. So we think that the person's more off task with the blue arrows and then on task with the red arrows, but you can see there's a one-to-one correspondence. So what we're predicting, they're validating. And this is just an N of 1, so this is really early on. Then we give them the intervention, and they tend to be more focused after that. But once we can identify optimal or suboptimal brain states, then we can be able to trigger just-in-time interventions, or we can trigger cognitive tasks that can then causally improve human performance. So I'll just give you an example of a study where you could do that. So let's say you want to like change repetitive negative thinking. What you would do is you have baseline assessments, and then you would have 3 sessions of neuroimaging. One would be, let's say, an induction task to look at which adjectives are related to repetitive negative thinking for that individual. That one could be a block design. That design would then identify a contrast image or voxels or regions of the brain that are corresponding to repetitive negative thinking for that individual. And then the second session would be to use real-time fMRI to track the state of that feature or network. And then you can trigger experience sampling. That will do 2 things. It will validate the association of the natural fluctuations of that signature with, say, rumination. And you can do the predictive modeling, like I just mentioned, to look at the data right before the experience sampling and build a person-specific predictive model that will identify the neural antecedents. You'll understand what the biological measures are right before the onset of that symptom. And then to understand causal mechanism, you do real-time neurofeedback to train people how to modulate that fingerprint. So that's just kind of an example of how you would do that. And then you can follow up, because you can do this with EEG, fMRI, you get the electrophysiological correlates of that network that you're trying to modulate. You couple that with EMA, ecological momentary assessment, as well as digital phenotyping. So you have 3 different inputs, and you can track people longitudinally that way. I'm just gonna do one. I really am running out of time. Here. I'll just say, you may question, if you're tracking the state of a mind or a brain, let's say you want to trigger a task, can you change their performance? Well, we've shown that you can look at, say, the default mode network here and PCC and SMA, and you can trigger stimuli to, say, sustain a vigilance task. When you present stimuli, when they're in a good versus a bad state, they get better reaction time when you are in a good state. Same thing with subsequent memory. If you present scenes to somebody when they're in PPAs in a better state, they subsequently remember better. So all of that is to say that if you ultimately we want to derive these multimodal measures, which could be EEG, that's an electrophysiological correlate of the fMRI, as well as heart rate variability, respiration, all of the things that you can measure in the scanner. But once you get that scalable version of it, then you take that and you can track people and identify when individuals may be about to fluctuate in various stages. Say you can detect when somebody's going to be vulnerable or somebody's going to be in a good state. And then you can trigger biologically just-in-time intervention. So you can track them with all of these other digital phenotyping measures and say, aha, you're looking at the data between the brain and the heart rate and the mind-body association. You can say, oh, this person's in a good state of mind or a bad state of mind. And if they're in a vulnerable state of mind, then you can trigger an intervention, which may be even specific to the person and the severity of the intervention. And then if they're in a good state of mind, you can even suggest, not only give them positive feedback, but you can suggest that they practice things like mindfulness when their mind is very capable of receiving that. And you could even suggest flow states, like if you're performer, you can look at mindfulness. This triggered a way to actually, get into the flow state. So I'll just close by saying we're hoping to develop, we'd love to collaborate with you on this mind balance training platform where the mind scope would be identifying these neural features that are associated with brain fluctuations, capture the electrophysiological correlate, couple that with, wearables, and then trigger just-in-time interventions or flow states until the person knows when that is going to happen and can modulate their brain networks apparently associated with it. So we're tracking all of these different directions with everybody here at MGH. And again, like I said, they started the talk saying I'm incredibly grateful and tremendously grateful for all of the work that you're doing here and for all the collaborations that we have ongoing and the collaborations that will come. So thank you very much.